Hi, welcome to Hard-Boiled Synthesis, Lecture 17. Oh boy, we are going to have some fun today. Now we're getting our hands dirty for real, where we're beginning to start synthesizing all the effects that we extracted from the studies. We're talking about 200 some effect sizes. Now let's play with these things. Let's analyze the overall effect. And again, remember, oh boy, the overall point of the course is to answer the claim of whether or not catnip is 10 times more effective than DEET. We are super close now. Mega close on answering that question. But before we start, we need to start really now talking about meta-analysis. And what is the point of this tool? And so now we need to start talking about stats and some of the mechanics and the functionality of meta-analysis. Now, in the theme of the course is I want to avoid theory as much as possible. We've gone through 17 lectures already where I'm just showing you how to do a meta-analysis. Now, the meta-analysis part involves many models. It's a modeling tool. And so I think maybe before we actually go to try to answer the claim, I'm going to save it for the next class. Right now I'm just going to show you with a bare bones, bare minimum, fundamental model of meta-analysis, which is essentially a weighted regression, or what we call as a fixed effect model. Nobody really uses a fixed effect model to synthesize their effect sizes. But it's the base coat before we start doing more fancy modeling techniques, right? So and so it's very rare now to actually see a meta-analysis published where they're just describing a fixed effect model. More typically now is researchers use what we know as a random effects model, and that tries to model details about how we think effect sizes are distributed and sources of variability associated with that. But that's not the point of today. Today's just to show you the bare bones, bare minimum, the simplest model we got to synthesize data, which is the weighted model, the weighted average. Now, I'm going to simulate some data, and then we're going to do a fixed effect meta-analysis in Excel. Why not? I got it all coded in Excel already, so we're just going to keep using those tools. But to do the more fancy modeling, we need to start using R. And so this will be like a warm-up for me to start using R again to do analyses. And so let's just begin, all right? I don't know how else to describe that we need to start making sense of all our effect sizes. Right, so we got a whole bunch of hedges D's, and we estimated their variances of each individual effect size. How do we make use of that? How do we analyze these study outcomes, these abstractions of study outcomes? Now, the first pass at it would be, well, let's just take an average of all the effect sizes, right? Like we got a huge column of effects. Why don't we just flat out take an average? I mean, yeah, super easy. Okay, in Excel you do average, zip up the entire column, boom. The average hedges the effect size across 226 effects is minus 0.26. Indicating, as a first pass, that catnip is not more effective than DEET as a repellent. DEET performs better at repelling mosquitoes than catnip. Now we don't use just a straight up average in meta-analysis because there's a lot of issues that make this value right here useful in terms of context, in terms of what we understand about each study. Not all effects are the same. Not all effects have the same quality. And one of the easiest ways to model quality is to think about how they are sensitive to sampling error. The number of replications a study uses really impacts how strong their test is into identifying an effect. And with the catnip studies, oh boy, I mean, these are heavily underpowered studies. Underpowered in a sense that they use very few replicates in their experiments. Ideally, under ideal conditions, you'd use as many replicates as you can, right? So you do a catnip experiment with hundreds of people sticking their arms in boxes with catnip and with DEET, doing that over and over and over and over and over and over and over again. And then you average all that stuff, and then you get a fairly good idea of the overall effectiveness of catnip. But we don't have that. We don't have the benefit of one study doing that experiment many times. What we have instead is many studies replicating that test. And so that's really what the power of meta-analysis is that we're treating each study as a replication of a test that aims to identify whether or not catnip is an effective repellent. And so this is one of the reasons why we don't quite do a straight up average of effects across study, is we know that each study differs slightly because of sampling error. Some studies may use many replications, some studies may use very few. We need to figure out a way to account for that variability to improve our estimate, to improve our average across all studies. And that's what the weighted average is. The weighted average is we weight by the variance of the effect size. The reciprocal variance becomes the weight for each individual effect. Okay? All this seems like mishmash, me talking to you, so maybe the easiest way to describe what we're doing here and why it's necessary is just to do a quick simulation so that you can visualize like, oh wow, okay, sampling error is a big deal and we should try to account for that in our models. So let me jump into R here. Okay, so my goal here, I'm gonna make things super simple here. Okay, I just want to show you guys sampling error and why we weight by the reciprocal variance of each effect size to get a better estimate of the average effect, right? We're not doing just a raw average. We're doing a weighted average because effects differ just because of the number of replications that they use. So my goal here is just to show you guys the variability that could emerge because of small sample sizes. And then I'm going to jump back into Excel and we're just going to do a quick fixed effect meta-analysis, which is a weighted average, making use of those reciprocal variances to get a better estimate of the mean. And so here I'm just going to really do things simply as possible. I'm not really going to tell you exactly what's happening on the back end, other than my goal is to simulate effect sizes that were estimated from experiments with small sample sizes. And then we're just going to look at what kind of effects emerge just by manipulating the number of replications in a study. Okay, so hypothetically, all studies are the same, right? Everyone's sticking catnip and DEET into their cages. Everything is the exact same. Same humans in each study, same mosquito species, same exact repellency concentrations. The only thing that differs is the number of humans used in that experiment. 3 humans, right? Sample size of 3, or 10 humans, sample size of 10. And then we're going to see what the overall effect that emerges from that variability. So I'm going to load in a library Metagear here just to make things simpler for me. It has some tools to simulate data. That's all it does. And so the first goal I want to do now is simulate variable sample sizes, right? So I'm simulating number of replications per study. Sample sizes, they tend to have like a Poisson negative binomial distribution. They're just random counts. Counts have this distribution, but I want to simulate it like a real diversity of sample sizes you would see with meta-analysis. And so typically you see many studies with few sample sizes, and then you have a few with large sample sizes. There's like a tail to it. And so so I want to create a bunch of random sample sizes, and random n. N is the word we use for sample sizes. Let me look at the, okay, so then we want how many random sample sizes? I'm going to simulate 500 studies. The average sample size is going to be small. So most studies, I don't know, we could say for the repellency studies, right? There was not one repellency study that had a sample size greater than 10. I mean, all of these studies are they are using very few replications. That means they're very sensitive to sampling error. You'll see here. And so I'll pretend as though the average sample size is around 6, right? So we're going to replicate 500 studies, and on average, they're going to have a sample size of 6. And then here's where it gets kind of weird, is the negative binomial stuff that I'm going to simulate. And I'm just going to throw in some random fraction there. That's just to indicate that there's a long tail to the distribution. And then I think I want to plot this, so just so you could see. And I think it's just hist. So sample sizes, and then I want, since we have a lot, we could just break things down, bin things in many different ways. Okay, so just to recap, 500 random sample sizes. Okay, what I'm trying to do is simulate studies with small sample sizes and some with many big sample sizes, and then we just plot that as a histogram. I think I could just source this. There we go. Let me double check to see if you actually see this plot here. You do. That's a win. Okay, so as we tried to do, we got a bunch of many studies, mostly have small sample sizes, less than 20, and then we have a long tail of studies that have large sample sizes. This is the typical distribution of what we would see in a meta-analysis. You have 100 studies. They all vary in the number of replications they use. Some have a lot. Most have very few. Okay, so the next goal is to use those replications to simulate study outcomes. And for the sake of simplicity, I'm just going to simulate outcomes as correlations. That's just another effect size. So, so I'm going to create a bunch of random effect sizes. They're going to be correlations. R is just another way to describe a Pearson correlation coefficient. I want to simulate an average correlation of 0. And I want each one of these correlations to be estimated by our random sample sizes. I think this is all I need. I forgot an equal here. There we go. All right. So now I'm taking those sample sizes that we randomly generated and now modeling a bunch of random effects based on those sample sizes. The only thing that varies across all these effect sizes is the number of replicants used to estimate the effect. Okay, and the estimated effect here is 0. So the goal would be, if we would do a meta-analysis, to estimate this unknown effect. And if we do things right, the unknown effect would be 0, like we modeled it here. Effect sizes, random. Okay, so I need to plot this. So we're going to plot the sample sizes. Which will be on the y-axis. And then we're going to plot the effect sizes, which will be on the x. No. This will be on the x-axis. We want the sample sizes to be at the bottom, and we want the effect sizes to be on the y-axis. Now I'm going to change the color of these effect sizes to make things just look nice, but you'll see later that when I modify this thing, it'll make my points much more clear, maybe. I don't know how else to say it. And I think all I'm going to do is change the color. So red, blue, green, and I think it's super easy to make things look cool. And then finally we add an alpha value. So all I'm doing is just changing the color of each effect when I plot it. And I think that is it. We don't want anything else than this. And then I want to add a line showing the null effect. 0. I think this is everything we need. I'm going to not plot the histogram anymore. Plot everything we need to show the relationship between the number of replicates versus the estimated effect. And you'll see that with fewer effects, there's going to be huge variability in effect sizes because of sampling error. That's what's generating the heterogeneity across outcomes, is poor replication. Okay, so and I don't really know if this is gonna work. Let me just bring up that window. Error. Red. Oh yeah, it's red, green, blue. Woof. Never easy. Bring that up. Boom. There we go. Is that it? That's it. Okay. Let me check to see if you guys see this. You do see it. Okay, so that is essentially sampling error. This cloud of, like this funnel cloud of what you see is all due to sampling error. Studies with few small sample sizes can over or underestimate the effect heavily, but with more replicates, they converge on the true underlying effect, which here was simulated as 0. Right? So the studies with many sample sizes, like hundreds, are really close to 0, but studies with fewer than 20, man, they're all over the place. And since this is a correlation, it could range from 1 to -1, and we see that entire range, right? So a single study with like 4 replicates, the estimated effect could be all over the place. This is why we don't take a straight up average of these things, because we know the studies with the small sample sizes is not really estimating the effect in a high precision, high quality way, right? There's no way it can, because it's really sensitive to sampling error. And so if we could downweight the contribution of studies with small replications, then we could improve the way we estimate the predicted effect. Here is 0. And so I'm just going to change the colors on this, so just so you could see how a weighting scheme would impact the contribution of each effect size when we take a weighted average. And so I think all that means is I'm going to modify the colors a bit in the way this is plotted. Now I need to calculate some weights. And the weights is, again, the reciprocal of the variance. And here I'm just going to assume the variance is the sample size. This is not what is happening with a meta-analysis, okay? Whenever someone says, I'm weighting by sample size with meta-analysis, that's not what they're doing. There's no meta-analysis out there that just weights strictly on meta-analysis. Some meta-analyses make shortcuts and only weight by sample size, but you're overlooking really important sources of variability in the estimation of the variance. If you look at the variance of Hedges D, there's more than just sample sizes included in that. D is included in the estimation of the variance. And that's, again, because D has a non-central t distribution. D, including D in the variance estimation, is important to properly model the variability of the effect size, right? So if you just use the sample sizes, you're overlooking key aspects of how we model effect sizes. And each effect size has a different way to estimate the variance. And sample sizes are important for that, but there's always a little nugget included in those equations that helps improve the variance estimate associated with the effect. But here I'm going to simplify it. I'm just going to use the sample size, the inverse sample sizes. Right, so this is the reciprocal weight. So I took the inverse of the sample size. And maybe I want to do a bit more than that. I think what I want to do too is just for plotting purposes, throwing in a bunch of variances in there is not beautiful for plotting. And so this addition, I'm basically adjusting all the weights so that they sum to 1. And that's just for plotting purposes. Don't ignore this. This is not the meta-analysis part. And then I'm going to throw in the weights here. So the first part is the red. So now every weight, every effect size that is going to be down-weighted in a meta-analysis is going to get colored red, right? So their overall contribution to the average will be lessened, right? We still want to include them. They include important information, but we just don't want them to have a high impact when it comes to averaging the effects. Hopefully this works. I feel like I messed up at some point. And then finally, nope. Yeah, I messed up here somehow. Let me just look at these weights quickly. It's never straightforward. Maybe if I do this instead. Ugh. Okay, I'm going to stop. If this doesn't work, I'm just going to continue. So this is all unnecessary random walk on how to describe things for you guys that I don't need to do. Wait, let me just change this for a second. Maybe I need to normalize those weights again. Again, this is just for plotting purposes. I want to make the visualization that the studies with the small sample sizes get down-weighted in a meta-analysis. Their overall contribution gets decreased because they provide information, but they don't provide information we trust in estimating the overall effect. Yes, there we go. Okay, so I got the colors backward, but whatever. So everything in blue here are studies that are that get down-weighted, right? The overall contribution becomes less impactful when you take the average. An average, just a straight up regular old-fashioned average, assumes that every effect has equal contribution. In a weighted average model, now we're assuming that each effect does not have equal contributions. Now we've adjusted the contribution of each effect based on how sensitive they are to sampling error. So in this case here, studies with high replications get weighted heavily. Things with super low replications have a much smaller weight. Much smaller contribution to the overall average effect. This is a fixed effect meta-analysis. This is what we do. We compute weights for each effect size, and then we downweight individual studies based on this estimate of sampling error. That's the first key part of meta-analysis, is this weighting. More elaborate models, like the random effects models, does not fully assume this model. This model assumes that there's a single underlying effect that every study tries to estimate, right? Just like the way I modeled here. The single underlying effect here is 0. And every one of these effect sizes is trying to estimate that 0, but sampling error prevents each individual study from precisely estimating that effect. The better model is to assume a random effects model, where this underlying effect is not fixed, but random. It has a distribution. It's a mean with a distribution. And so if I was simulating a random effects model, every single one of these effects would not be estimating a single underlying effect, but will be trying to estimate a population of effects, right? So now we've increased the hierarchical structure of the data, where before we assumed that there was just one true underlying effect, but a random effects model assumes that there is population of effects that we're trying to estimate. That's a more realistic model. Ecological studies are incredibly variable. We don't know what the underlying effects are, whether they're diverse, whether there's many effects, and so we need that baked in, that additional source of variability into our models, so that we don't incorrectly identify effects that don't exist. And when we assume this random effects model, basically what we're doing is adding more variability into the model. And so this kind of tends to make our overall assessments more conservative, right? If there's huge variability across studies, we want that baked into our model. This model right here, this fixed effect, does not account for that variability at all. The only source of variability is sampling error. And so this is not ideal for what we want. Just for fun, let me just throw in, let me change the alpha values of this model just so you get an idea of further emphasis on the weighting. I don't know why I'm obsessed with trying to show you guys the weighting of these things. Let's see if this works. Oh, got it backwards. Okay, so what I'm trying to do is like, see, this is totally backwards. So here, this is the inverse weighting. So it's studies with large sample sizes are almost negligible in the analyses. This is the opposite. So what I want is to make down-weighted studies more invisible, more transparent, to emphasize their decreased contribution. This is all exciting stuff. I don't know why I'm doing this. There we go. Okay, so it's not that quite clear, but you could see that there's like an increased fuzziness near the end of, near the small sample sizes, right? So this part here is fuzzy, and that's because each individual study has a less impact in estimating the overall effect. We still include them, right? They do have information. This is a distribution. We want full representation across this distribution. But now we're just deciding, hey, you know what, these studies with few replicates, they're not really adequately estimating the effect. Let's downweight their influence, emphasizing studies with more replications. Random effects model, though, the true meta-analysis model, we don't quite make that call so rigid-like. All right, one last simulation just for fun, and let's try to simulate the repellency studies, right? This last simulation, you know, I simulated studies with like hundreds of samples, replications, sample sizes of 100. There was not one of those in the repellency studies. The maximum was 10. So let's have an idea, let's get an idea of the weighting involved in when all your studies have just are highly sensitive to sampling error. How am I going to do this? I need to change this random sample sizes. So now I'm going to take the mean to be on average 3, and then I'm going to get rid of that tail. Right? So there's no more studies with high sample sizes. Now everything's just going to be small sample sizes. Rerun that, run the weights. Okay, let's see if this works. There we go. Okay, so the only one study here had a sample size of 20. The majority have really low sample sizes, under 10. And you can see the effects of the weighting here. So all this end here is fuzzy, is bluish. These are all the studies that are going to get down weighted when we take an average across all these effect sizes, right? So the goal is to estimate this. And each one of these points is a replication, is a study that tries to estimate this. But they can't individually, they can't do it because they have too few replicates. That's the whole point of meta-analysis, is to treat each study as a replication to try to estimate that overall effect. All right, let's do that with our effect sizes. This is fairly quick, easy to do a... Let me close this, jump into Excel. It's fairly easy to do a weighted average, using old-fashioned meta-analysis fixed effect. Remember, all these tools were developed in a time when people didn't have fancy computers, and so we needed a way to quickly estimate these things. And so we're going to use like this classic ANOVA style, least squares, just sums of squares types of analyses to get the weighted average. Okay, so we already took the raw average. And now we want to individually weight each Hedges D effect size by the reciprocal of the variance, the inverse of the variance. We estimated the D, we got the variance of the D, that becomes the weight when we inverse it. So the first thing, I'm just going to follow Hedges ANOVA style notation here. We're going to do TW, which is the total weight. I don't know. It's just the inverse of the variance. Okay, that's the first part that we're going to calculate. Second part is we're going to do the total weight of Hedges D. And I think the equation for that is just D divided by its sample size, its variance. And then finally, we want the square root of that. And so I think that is just the square root of D divided again by the variance. Oops. And we call this the TD squared. TDW squared. TWD squared. Right, so we got the weights, reciprocal weights, inverse weights. We got the Hedges D divided by the variance. And then we have the squared of the Hedges D divided by the variance. This is all we need to estimate the fixed effect meta-analysis. Okay, so now we want the sums of these things. So I'm just going to take the sum of all the weights. The sum of all the weighted Hedges Ds. And the sum of all the squared weighted Hedges Ds. Okay, so this right here is what we're going to use to estimate the weighted average. And I think the weighted average, I need to look up the equation. I think it's just the TWD divided by TW. So I'm just going to do the weighted average here. Right, so the TWD, oops, wrong one, TWD divided by, there we go. So the raw average really overestimated the effect, that DEET is doing way better than catnip. Doing the weighted average, accounting for some of the sampling error, the effect is actually almost half that, right? And so DEET, when you account for the sampling error across studies, is not doing that much more better than catnip. Right, so this is just like, right? fixed effect meta-analysis. Right there. Okay. So just to, again, we're just learning about the models for meta-analysis here. This is not what the overall effect will be once we do the true meta-analysis, because we're going to assume a slightly different model, the random effects model. But this is what we got right now if we account for sampling error. I mean, okay, that's it. So they, just on using raw averages, catnip is not 10 times better than DEET. DEET seems to perform better on average. Accounting for the variability associated with each studies, DEET is still performing better on average than catnip. But a big chunk of that was because studies were undersampled. Okay, so I'm going to end it by this. Next lecture, we're actually going to truly answer the claim of whether or not catnip is 10 times more effective than DEET, because we're going to use the real meta-analysis model. Today, I was just talking about weighting, sampling error, but the true meta-analysis, we take it to a whole different level. We think about effect sizes at more of a hierarchical distribution level. All right, this is getting exciting. We're getting somewhere. Tomorrow will be the day where we answer the claim. I hope this is useful. Oh boy, take it easy.